Hello, in this video we will discuss about the immune system in plant. So what is the immune system? Let's begin to understand. Here is the plant, and this plant is contain a cells. There are different type of cells: mesophyll cell, epidermis cell, vascular cambium, core cambium, xylem and phloem. There are different type of cells. So here I am drawing just a general structure of the cell for understanding. So here is the cell wall, here is the mitochondria and chloroplast, which that is used for the photosynthesis. Nucleus is used for the storage of the genetic material, and mitochondria is used for the power energy production. So that is why it is known as the powerhouse of energy. So on the other hand, here is the vacuole that is used for the storage mechanism. So the ions and water will be stored in it. And on the other hand, here is the peroxisome, mesosome, and glyoxysome are present, which that is a little organelle. So here let's begin to understand after the invagination through cell wall, the fungal oomycete, for example, Hastoria will move invaginate inside, and on the other hand, the aphids, which that is invaginate through stylets and nematoda, and this stylet will move inside to the cell intracellular into the cytoplasm, and after that into the cytoplasm the recognition will be occurred through plant cell, and this bacteria is also recognize the basically is the bacterial recognition receptor after the pathogen recognition receptor. And in this way here is the produce the different type of molecule also. This is the pathogen recognition receptor and the pathogen death domain receptor. So on the other hand, the pathogen associated molecular pattern and death associated molecular pattern, all these trigger the effector here, but here is the damp and PAMP, which that will lead to produce the pathogen trigger immunity, while here is the effector of these through a bigger organism, which that is the effector will bind with the node like receptor, and after the intracellular node like receptor is here. This is two type here, and three type is unknown. So three type can be, but I am not drawing here. So here is the effector decoy containing protein and effector another protein in the intracellular node like receptor is the basically here. And in this way the effector will bind after the binding with the effector to activation this node like receptor, and this node like receptor will trigger after the activation to produce the effector trigger immunity, and this effector trigger immunity will be phosphorylase. The other transcriptional factor will bind with the DNA of the host, and this immune gene expression will be occur against the pathogen for defense mechanism. And in this way here is the other node-like receptor is the sensing effector mediated alteration in decoy protein that structurally mimic an effector target. So here is the effector target for that. So it will be target to the effector for the killing of the effector. Effector means the pathogen. So here is the prototypical pathogen. So the effectors you know about that is the pathogen associated signal. So here is the different type of the pathogen. We will discuss about the prototypical pathogens first. This prototypical pathogen is the patterns which that is the death associated molecular pattern will be bound with the death domain recognition receptor. After the death domain recognition receptor, the activation of the defense activation of defense, and in this way the transcriptional factor will bind with the signaling defensin. This defensin will produce and defense against the pathogen. So this is the DNA of the cell and the nucleus. So this is the pathogen, and on the other hand, the broad host range necrotrophic pathogen. So the broad host range necrotrophic mean the broad mean the, for example, the fungi which that is dispersed throughout the cells. So in this way, here is the VAC1, which that trigger this toxin acid, and other weeds which that toxin to activation of defense signaling again these things. While the when direct, the toxin will move into the cell, so it will be inhibit the activation of defense signaling. While here is the host specific necrotrophic, which that is the host selective toxin, which that can be directly detoxification non-recognition, while LAEO is the basically the virulence target, and this is the R protein. This is the R protein, and this R protein will trigger the necrosis, and this virulence target will trigger the R protein, and the necrosis will be occurred, and in this way the effector trigger system. So on the other hand, here is the TT1, which that trigger and to produce this thing, while the hemibiotrophs is also used for the effector and bind with the R protein to activation of defense signaling. So here let's begin to understand important things, the iron associated immunity. So the iron deficiency will lead to increase the immune response. So here is the ferritin, defensin, disruption of pathogen, iron signaling. So in this way the pathogen, if the more iron, so it will decrease the cellular iron. So in this way here you can see, so the iron will move inside to increase the iron, but important thing is that here is the ATPase. This ATPase basically is pumped to out to ferrous to convert ferric to convert a ferrous, and this iron basically is contained, and this is the iron iron receptor iron transporting, which that is used to move the iron throughout the body. So here is the endodermis, cortex, epidermis, and phloem and xylem. So let's begin to understand with the leaf. So here is the, you can see the leaf, here is the iron are present inside, and here is the OPT, that is another things, and this mesophyll cell, and in this way here is the iron inside, and this is the basically is the vacuole and the mitochondria and another things which that transport the iron to other things, other part. When it will transport the iron, so the deficiency of the iron will be occur in the cell. So the deficiency will lead to the bacteria, fungi, pathogen response to kill them. How? So here is after the binding with the pathogen associated molecular pattern, the biotrophs. After the binding, the recognition by receptor and the recognition by receptor during the low iron will lead to increase the nitric oxide and ethylene and sialic acid. After the sialic acid and nitric oxide and ethylene will be more, will trigger the effector trigger. This is the basically the effector trigger, not it basically ethylene, but it is effector trigger, sorry. So in this way here is the reactive oxygen species, iron deficiency response, the coumarin biosynthesis, which that will lead to give the immune response and kill the pathogen, and here is the cell wall and reinforcement and systemic immune activation will be occur. And in this way here is the another the necrotrophic pathogen, basically is the effector trigger again with the jasmonic acid. After the jasmonic acid basically trigger with the effector trigger complex formation to reduce the pathogen growth, and this is the important things. And here the hypersensitive response lead to cell death. So in this way the hypersensitive response this was for lead to cell death due to the recognition through nitric oxide and the effector trigger bind and sialic acid, which that is used, and jasmonic acid is used for the cell pathogen growth will be inhibited. While here is the another pattern, which that is the effector delivery. After the effector delivery, when deliver the pathogen hyphae and the effector delivery bind with the R protein. After the binding with the R protein, the reception of the effector to basically the effector triggered immunity will lead to iron Fenton reaction, and this Fenton reaction will lead to produce the reactive oxygen species, but the glutathione which that will be inhibit, but effector trigger immunity will inhibit the glutathione, and this inhibition of the glutathione will trigger the Fenton reaction to lipid peroxidation, and the ferroapoptosis or cell death will be occurred due to the deficiency of the iron. So that is the mechanism, which that is when decrease the iron in the cell of the plant will lead to trigger the immunity for the killing of the pathogen or itself the apoptosis. So thanks.